So moving into session 3 now, our final session. We have some judges. Steve, do we have all the judges here? We ready to proceed, or how do you want to?
Yes, I think we have a number of them. We're still 1 or 2 coming in, so we'll
take a couple minutes.
Yeah, maybe take a couple minutes. We'll get it all.
Okay.
Away. Thanks.
Sounds great.
Great. Right. So Zach, you can put it on the. So hold music if you want. Matt can take a little break too.
Great.
We'll kick it off around in 3 minutes. Let's call that 12:58.
Perfect.
All right, I think we are about ready to begin. Have everybody. So Matt, I'll hand it back to you when you're ready.
Okay, great. So thank you so much, Steve. For those of you that may not have joined us for previous sessions, my name is Matt Bresnahan. I'm a partner at Wilson Sonsini in our life sciences practice and the moderator for today's life sciences track. We're about to start our third session for the day with another great set of early stage companies. And then as a programming note, after the session, at about an hour from now, we'll have a corporate panel followed by some remarks from Andy to close out the day. So definitely stick around for that. Okay, so format wise, and then I'll turn over to the judges to introduce themselves. Format will be the same as previous sessions. For anybody who wasn't able to join those, we'll have 3-minute pitches from each company, followed by about 9 minutes of Q&A from our judges, as well as anyone from the audience that would like to submit them through the chat log. We'll get to them, and I'll do the best I can. So far, reasonably good. I think if you ask Steve of keeping everybody on track, although he's not shy about letting me know if we go over, so I will pass that along if we do. All right, great. So what I'd like to do now is turn it over to our judges. I'll go through the list and then I'll have them each introduce themselves. So we have Marianne Vogelsang, we have Kwame Ulmer, we have hardest working man in show business, Ryan Witt, who's been with us all day. Thank you, Ryan. And Martin Brenna and Sean Aurora. So maybe Marianne, if we could have you introduce yourself first.
Actually, I think Marianne's still logging in. Sorry about that.
Oh, she is. Okay. Well, hopefully she will towards the end. Kwame, if you'd like to go. And you're on mute, Kwame.
Maybe frozen too.
All right. We'll go down the list. Ryan, you're on.
All right. For those who weren't here earlier, so I'm the hub director of UCLA's Medtech and Digital Health Innovation Hub, also a angel member, angel investor for the past probably 6 years, and been a member of Tech Coast Angels Los Angeles chapter for almost 3 or 4 years. For those who don't know Tech Coast Angels, I actually met Kwame back then there. They're a group of over 450 angel investors locally in Southern California. They very active. We invested over around $20 million last year in around 64 different companies. So very active group, and try to get back to you within 30 days. So very quickly.
Excellent.
Great to be here.
Thank you, Ryan. Martin, would you like to introduce yourself?
Sure. Good afternoon, everyone. I'm Martin Brenna. I am part of the due diligence team at Kairos Ventures. We are based out of Pasadena and Beverly Hills, and we are interested in life sciences companies and physical sciences companies, usually at the very early stages. It's great to be with you this afternoon.
Excellent. Well, thank you for being here, Martin. Sean?
Hi, everyone. I'm Sean Aurora with Mila Capital. We specialize in pre-seed companies. So like Zach mentioned, there's people who invest really early, like angels. We do that too. We're looking for founders. We try to help them get onto the VC rails. Normally there's something that generally PhDs don't come from the startup world and they need every edge possible. So we like to jump into very promising teams and help them get there. And we specialize in hardware technologies. So there's a lot of diagnostics companies that are about to pitch, and that's our core.
Excellent. Well, thank you, Sean, for joining us. Marianne, are you with us? Looks like you are.
Yes,
yeah,
I
would you
am.
like to introduce yourself?
Can you hear me?
Yep,
yes,
you can.
Hi, sorry that I had technical difficulties today.
No
my name is Marianne Vogelsang. I'm the Senior VP of Clinical Chemistry R&D at Curative. And I have background, I've built up clinical labs in the CLIA space. I've worked with FDA on some of our medical devices in both startup space. I've also spent the last couple of years at USC doing corporate collaborations. And so I'm excited to hear and provide.
Excellent. Thank you for joining us, Marianne. We're excited to have you. Kwame?
I'm Kwame Ulmer. I'm a venture partner in Wavemaker 360 Health. We're an early stage investment fund, and we invest in medical devices, digital health solutions, and diagnostic companies. It's a pleasure to be here.
Thank you, Kwame. We're excited to have you as well. So a great group of judges. I'm excited to hear from the companies as well as the judges. So why don't we get right into it? The first company we have scheduled for the third session here as we start is Sayenza Biosciences. And Derek, are you with us?
I'm here.
Excellent. Feel free to share your screen and start when you're ready.
Great, thank you. Did you know that fat, we're talking love handles, the inner thighs, or even that pesky double chin, is the largest source of stem cells in the body? Hi, I'm Dr. Derek Banyard, and I left plastic surgery residency to found Sayenza Biosciences, where we are harnessing this liquid gold for limitless aesthetic and regenerative medicine applications. I'm sure most of you are familiar with fat grafting to the butt for the Brazilian butt lift, but did you know that adipose stem cells are currently being explored for the regeneration of heart tissue after heart attack, the restoration of nerves after spinal cord injury, and the regeneration of bone after disfiguring cancer surgery, just to name a few? Problem is, how does one get from this liposuction canister to an injectable therapeutic? Traditionally, a surgeon would have to lug a centrifuge into the operating room where they would load fat into syringes and then wash, clean, and spin the fat. Now, there are newer devices on the market that facilitate this, but there still requires further downstream processing and filtration before one gets to this injectable therapeutic. This is all problematic because it's time consuming, requiring up to an hour of processing. There are multiple steps and manual input that increase the risk of contamination, and it distracts the surgeon from the patient because he or she is focused on the processing. At Sayenza Bio, we are developing a multifunctional patent-pending reusable platform that involves single-use disposable cartridges. It processes the fat in about 15 minutes and is fully automated, allowing the surgeon to focus on patient care as opposed to the processing. The platform we're developing offers unparalleled benefits in the market. The traditional fat processing method and some other simple devices are useful for fat grafting or transfer, and there are newer devices on the market that are designed to produce an injectable therapeutic. But Sayenza Bio is developing the industry's first platform that is multifunctional and useful for both. It's fully automated, carries minimal contamination risk, is reusable with disposable cartridges, avoids the use of harmful chemicals, reduces OR time and thus costs, and allows for the processing of both large and small volumes. I mentioned before the multiple applications that are exciting for the potential use of adipose stem cells, but we plan to enter the market in aesthetics, not only because it's valued at $6.25 billion, but it's cash-based, meaning no negotiations with CMS, insurance companies, or large care providers. We anticipate fairly quick FDA approval for our patent-pending Class 2 medical device via the 510(k) pathway. We will then begin clinical trials for various indications and seek regulatory approval for specific stem cell claims to extend our portfolio. I co-founded this company after leaving plastic surgery residency, where I saw firsthand the tremendous need for this technology. Dr. Jared Hahn is professor of biomedical engineering and an expert in processing and microfluidics. And Dr. Alan Widrow is a plastic surgeon, professor, and director at the Center for Tissue Engineering at UC Irvine, plastic surgery. And he also serves as chief medical officer at the fastest growing professional skincare company in the country. Dr. Siamak Aga is a Cambridge University trained Newport Beach plastic surgeon, and he serves as our clinical advisor. And we also understand the importance of regulatory affairs and quality assurance and have assembled a team of experts with over 75 years experience who have been involved in over 100 successful 510(k), IDE, and de novo applications. To date, we've raised about $95,000 in non-dilutive funding. We hope to receive a phase 1 SBIR grant later this year, which will pave the way for a phase 2 SBIR grant in 22, so that we can complete pilot device development and submit a 510(k) application by the end of 23. Thank you.
Great. Thank you, Derek. Hope we didn't lose our other. I see Ryan. Do we have our other judges as well?
Yes, so in the competitive landscape, you'll probably hear this company later today if you stay on. But Synova Life Sciences, have you heard of them? Kind of thoughts on. I think the market's definitely for aesthetics is big, but thoughts on and I'll ask later too for the on their technology.
Sure. So Synova, I don't want to speak out of place, but it's my understanding their technology is designed for the processing of adipose tissue to release cells for cellular therapeutics, cellular injectables. As I showed in our competitive landscape slide, not only do we operate in the cellular injectable space, but also the fat grafting space with an all-in-one device that is multifunctional and can obviate the need for input and output, if you will, from the users, whether that's the surgeon or the other technicians in the operating room.
Thank you.
Derek, it's great to see you again. If I remember correctly, you you had a use case for diabetic foot ulcers and at one point, and you pivoted to aesthetics. Can you help me understand what changed your thinking about your strategy to go from diabetic foot ulcers as one use case to aesthetics?
Yes, thank you for your question. And so we as a company still have a tremendous focus on meeting and treating health disparities. Our device will allow a very cost-effective means of stem cell therapeutics in low resource environments. However, to get to market, it's desirable to go after a market that is both easily accessible with low risk. And we believe aesthetics is the best way to get our foot in the door. One of the scientific findings of our technology has demonstrated that not only does our technology isolate stem, it also enriches and potentially activates stem cells that are specific to diabetic wound healing. But that is something that we're going to explore further down the road once we get our foot in the marketplace.
Thank you, Derek.
Derek, I'm guessing one of the benefits of that strategy is also an easier clearance path. Was that part of the decision?
100%, yes. So the 510K regulatory approval pathway is based on predicate devices of two devices that are in the market. One, which is a phase 3 clinical trial for the treatment of knee osteoarthritis. So that's an early market that we can enter. But in order to make claims for stem cell claims for wound healing, that would require more extensive regulatory scrutiny and clinical trials.
All right, thank you.
Well, I'll jump back in. Everybody's shy today. I had my cup of coffee. So Derek, did you say you expect clearance by the end of 2023 or to submit by the end of 2023?
The timeline we're working with is submission in 23, but a 6 to 12 month approval or turnaround.
So let me poke at that, Derek. I know this is a long game, but what would it take to compress that? What would you need to be able to submit your 510K in 2022?
Everything to go right.
Do you have a working prototype at this point?
So we have working prototypes of 3 out of 4 of our internal components. And the internal components are our cartridges, if you will. The platform itself is a simple peristaltic pump-based platform, which essentially we need to put into a box. Once we complete that final internal component, which we anticipate in the next 3 months, then we're going to start kind of putting everything in sequence to kind of flesh out our proof of concept so that we can then move on to a engineering firm to produce a commercially viable product. Now, it's certainly possible that could be done within the next 12 to 18 months. But as I said, everything would have to kind of go perfectly.
Thank you.
So Derek, you talked about the current process and the time it takes in the operating room and the physician having to do part of the process. What do you anticipate the cost would be for one isolation using your system versus the current system?
So one of the reasons we created this company and developed this product is because there's no standardized approach to the processing of stem cells. I mentioned there are 2 companies that are kind of in phase 3 clinical trials. One of the standards of care right now is the use of an enzyme called collagenase to isolate those stem cells. Mm-hmm. That process requires a technician to go to the operating room with the surgeon, which adds tremendous cost. It's a very timely procedure. It takes over an hour to isolate the stem cells. And, I don't know the cost of goods for said devices, but the cost, everyone knows the cost of a minute in the operating room is pretty significant. And so we anticipate that we would be selling our cartridges for less than $1,000. the potential reimbursement for a surgeon on any kind of liposuction and cellular injection is anywhere in the ballpark of $3,000 to $5,000. So we're looking at a pretty tremendous kind of ROI for the surgeon.
Got it. Thank you.
Great. Any other final comments or questions from any of the judges? Some great ones so far. Okay, Derek, well, great presentation. Congratulations on all the success and best of luck with the company.
Thank you.
Thank you. Next up, we have Stylo Sonic and Reza.
Hello.
Hey, Reza.
Can you hear me well?
We can.
Okay, let me share my screen.
Good to see you again.
Good to see you too. Okay. Let me know when you're ready.
Yep, feel free. Feel free to start when you're ready to go.
Hello, everyone. Thanks for the opportunity. This is Reza. I'm co-founder of StyloSonic. My dentist tells me not to pick my teeth with any sharp metal objects. Then as soon as I sit in the chair, she grabs a metal hook like this and starts stabbing my gum. In fact, your dentist needs to poke the gum around each single tooth 6 times to check whether your gum is healthy. This adds up to nearly 200 pokes per visit. Do you recall this painful experience? Well, if you don't, perhaps you should ask your dentist to pick up the metal hooks next time you go in, because half of Americans over 30 years old have some sort of periodontal disease, gum disease, which results in dental pain, tooth loss, and reduced quality of life. The current standard of care to detect this disease is a manual probing with this metal hook, which is inaccurate, subjective, and slow, not to mention a dreadful experience for patients. There have been some efforts to develop, to overcome the limitation of manual probing by developing digital probes, which are still invasive and inaccurate. We introduced GingiWatch, a novel imaging solution for periodontal evaluation. It is a poke-free and painless solution. GingiWatch collects key periodontal metrics in one quick scan, which is a non-invasive, accurate, and repeatable measurement. GingiWatch has a pen-sized handpiece that performs soft tissue imaging as it is moved over your gum by your dentists or hygienist. GingiWatch saves chair time up to 80% during diagnostics. Unlike manual probing, it is a 1% job. Our solution assists dentists to detect early stage gum disease. Painless GingiWatch process makes patients happier and prevent costly late stage treatment. Our sustainable competitive advantage is our intellectual property, as the StyloSonic co-founders are the inventors of this technology. Our total market opportunity, our customers are more than 200 dentists in the US. Interestingly, there are more than 10 times dental office in the US than Starbucks stores. Our total market opportunity is about $8 billion, which is based on a fixed revenue of unit sales and recurring revenue of consumable and software subscription. GingiWatch is a Class 2 medical device with 510(k) regulatory pass. It will take 5 years and about $10 million to launch GingiWatch. We are projecting to have over $120 million after 5 years of launch. StyloSonic teams include expert leaders in imaging instrumentation, as well as a strong advisory team to help the clinical study and grow the business. We have a unique technology with the team that can deliver it from benchtop to chair side. We have already secured $300,000 in non-dilutive funding from SBIR Phase 1 and National I Corps, and we are raising another $700,000 to move forward. Please join StyloSonic to save teeth and lives. Thank you for the opportunity.
Great. Thank you, Reza.
Of course.
Let's let the judges come back on here, and then we'll turn over to them for anybody that would like to lead with the first comment or question.
Sure. Reza, great job on the presentation.
Thank you.
Admire what you're building. It's a really hard thing to build because right now there's a status quo product that's really, really cheap. And you're trying to bring out a product that has some sort of patient benefit, but it's going to be more expensive for the dentist. How do you think you're going to sell this to dentists?
So there are a lot of benefits to dentists as well. As I mentioned, the very first one, the dentist, as we have been interviewing over 50 dentists and periodontists so far, they have a difficulty of diagnostic this disease. And the biggest problem of doing the diagnostic with this manual probe is this is very interoperative dependence. So one hygienist measure different numbers of pocket depths and in general diagnostic from the other. And as we talk to dentists, it's a very burdensome for them because they even cannot trust the measurement if the pocket depth is over 5 millimeter. So they repeat the measurement. So it saves a lot of time upfront, as well as I mentioned, this manual probing, if you have the experience, someone is probing, another person is writing the numbers, even though this is regardless of the non-accurate process. And then at the end, when the dentist showing the results to the patient that like this teeth needs more care and we need to do like bone graft or anything at earlier stage instead of implant at later stage, it's gonna make them more revenue at the front end instead of back end. At the end, because you want to compare the price, our device is gonna cost $29,000 based on our evaluation right now and our cost base. And if you want to compare with the current intraoral scanners in the market, which dentists are using, and the range of the price for the intraoral scanners are $30,000 to $50,000. And these intraoral scanners just make a 3D map of your tooth for the orthodontist and planning for implant. But our device is imaging beyond the surface and showing the depths without any invasive process. So, and with the quick calculation, and another thing which is important is the reimbursement process. They are already CPT code that dentists can use to charge the patient, and the range of the charging is between $80 to $150 that our device stays there. And if you just use the very lowest range of $80, with just seeing $15,000 patient per year, which is on the very, very conservative range, because based on the data out there, every dentist sees over 3,000 patients per year. And this has been the data over the last 20 years. We are being very conservative with 1,500 patients per year. The rate of return and ROI is 4x when they are paying only $29,000 to our device. So it's a lot of benefits, plus the ROI is very, very impressive.
Thank you.
Reza, thank you for your presentation. I love your energy, first off.
Thank you. Thank you.
Yeah, a question, you said something just now in your answer that kind of piqued my ears up. You said, $30K to $50K for these intraoral scanners, and they mostly are helping for kind of implants. Is your scanner also going to fulfill the implants? And if not, one suggestion that I might have is maybe incorporating that into the device, because then you're going to hit the minimum and then better. So maybe then, that could be, I could see that as a very strong go-to-market.
Thanks for mentioning that. And actually, we have implemented a camera inside our handpiece. We are not just talking about that because it's not our focus to take the market out of them, but we have the capability of already implanted into our handpiece.
Can you just say that again? So will this also do the scanning for implant planning, your device?
Yes. Yeah, because we have included the camera, and the camera has two advantages. One of the advantage of the camera, they can show the patient, see like this side of your gum at the end is more red and it's inflamed. So they can show the patient as well as they can do the implant. Another good point about talking about implant, right now when they do the implant, this gum and implant attraction. So they use after implant, they cannot use this because this hook gets into the pitch of the implant. They cannot measure if the depth is good and the attachment is good. The good point of our device, it can measure the pocket depth next to implant, which is not impossible today.
Reza, you said you talked about to 50 dentists or so. When you go with the value proposition for this, what do you hear back? What is it that they are excited about? What are they maybe not excited about? What are the comments that you got from this kind of market research?
Sure. So these 50 interviews we've done, it was for regional I-Corp. At the end of this month, we are going for national I-Corp at the 28th of June, and for more 100 dentists and dentists to talk to. But the dentists we talked to so far, they're very excited about the quick process, because right now, if you go to dentists, some of you may experience this probing process takes 1 or 2 minutes. And the reason is that they are just sampling few of your teeth. And the problem of periodontal disease is maybe you have a deep pocket at one of your tooth and nothing else is a problem. And if the hygienist is missing that, that's a big problem. So the dentist we talk to, and if they want to do the whole mouth, it takes over 20 minutes because, as I mentioned, each tooth, they need to do 3 points inside and 3 points outside. And also they need to look for inflammation, which is right now the only way to see the inflammation when the gum is red. And when the gum is red, the inflammation is at later stage. So that's the only way to detect that. So they were talking about this quick process. Our imaging the whole mouth takes 4 minutes compared to over 20 minutes of current manual probing. And the second thing is they can monitor the disease. Right now, when they treat the patient, they use the manual probing the very first day they do the treatment, but the patient needs to come back after 8 weeks. And this after 8 weeks, if they use the probing, which is they are delaminating the gum from the teeth, which is against what they want to do. So they just sample few, but with our device, they can do the imaging now. And in 8 weeks, they again do the imaging without any invasion to the gum. They can overlay the results and compare if the periodontal disease and pocket depths have been degraded and it's in the right way for the health of the patient. But the problem we encountered, and all of us know the pushback on the price, some of the dentists, they said we have intraoral scanners and we pay $30,000, and some of them, like on the top level, they didn't mind. But in the middle level, they said 29,000 is a lot. But after we were talking to them about all this reimbursement and the existing CDT code, they obviously are aware of that. It's easier to talk to, but if you want to just talk to them, yeah, this is capital investment of $29,000. Of course, it's a lot of money for them comparing to this, but there are side benefits that we need to talk to. And we haven't had a lot of pushbacks, just they want to see if what we promise it's going to do it.
Got it. Thanks.
Sure.
I have a quick question. Are you presenting an opportunity in developing a replacement for an intraoral scanner, or are you developing a $29,000 replacement for a manual probe, or both?
Yes. So our first, basically, as I mentioned, we have the hardware of camera inside the scanner, so we can do both at the same time. But at the beginning, what our focus is to replace this standard of care, which is century-old process, which is ridiculous and it is unbelievable.
And what will you sell that for? You won't sell that for $29,000, or will you?
Yes, the camera is already still there. So yeah, the camera is still there, but we are not selling that just for replacing the intraoral scanners. Our push is more toward periodontal disease, which is the bigger problem in the community.
Great. So we're approaching time. Yeah, Marianne, do you have a question?
Yeah, I just had one quick question as far as accuracy against the technology.
Sure.
The standard technology with the probe. What is your accuracy with the acoustic imaging? I imagine that's what the probe with the acoustic end is what's giving you your pockets and your depth measurements. What's the sort of the specificity and accuracy that you're getting against the probe?
Yeah, that's really good to ask. Currently, the accuracy with the manual probing is plus minus 1 to 2 millimeters, because there are only millimeter size on this pocket depth measurement. Our device, because it's ultrasound imaging, we are in the range of 10 micron measurement depth. And we can, another good point is we can image the entire pocket depths. It's not just the sampling point. And even if one of the teeth, half of the pocket is deep, we can capture that because it's continuous measurement over the whole contour of pocket depth.
And then do you have any business model around the data that you'll be selling or that the doctor will have?
Yes, so our business model for software, at the beginning, our cloud-based software is to store the data. Basically, our handpiece, we have a cover sheet to give less hassle for the hygienist. It's a one-time use cover sheet they put on the handpiece. As I said, it's a pen size. They do the imaging and they throw out that cover sheet. The cover sheet has a barcode that the plan is to scan the barcode, open up a space in our cloud base. So all the data goes to cloud base and all the processing happens in cloud base to decrease the price of processing, because processing is very huge. The processing happens in cloud and instantly data would be available. At the beginning, we only have a $2,000 annual cost for the software and this processing. We have another AI-enabled assisting for the dentist. After a year or two of when we have a lot of data, we're going to implant another revenue streams, which helps dentists to pinpoint on the points that they need to take care of. It's assisting, it's not the diagnostic, just this assisting process, AI enabled.
Great. Well,
Thank
excellent
you.
presentation, Reza.
Sure.
Great feedback from the judges. I
Thank
feel
you.
like Steve's gonna barge into my house and with a cane and rip us off. But last comment I'll just leave you with from the audience to consider collaborating with cardiologists for heart disease prevention with the
Yes.
connection with gingivitis there, which you've
Yes.
probably thought about. So great job, Reza. Great to see you again. And
Thank you.
Congratulations on the success and best of luck.
Thank you.
Next up, we have Synova Life Sciences and John Chi. Hey, John.
Hey guys, nice to see you all. Nice to see some familiar faces. Derek, thanks for warming everybody up. I'm going to share my screen. Can you guys all see? All right, awesome.
Yes.
So I just want to start off by saying that I want to be 1,000 years old and playing basketball with my friends and hanging out with my family. And stem cells are one of the keys to being able to do this, and specifically my own stem cells. Now, fat is an incredibly rich source of stem cells. People typically have 500 to 2,500 times more of their own stem cells in their fat than anywhere else in their bodies. But these cells are trapped in their fat. So at Synova, we've developed the world's fastest cell processing system that can release millions of these cells from less than 2 ounces of harvested fat in under 3 minutes. The way most people do it now is with enzymes. It takes a lot of steps, has many potential points of contamination and error, and can harm the cells. So we've simplified everything to a single button push. We've reduced the time it currently takes from over 100 minutes to less than 3 minutes, and we've increased cell yields by 1.5 to 2.5 times with no chemicals or enzymes, with the sterility maintained in a single-use disposable cartridge. We get more cells and they're healthier cells. So it's a broad cell therapy platform that's already been used in point-of-care orthopedics for osteoarthritis, joint injuries, and joint pain, in skin and hair regeneration. We're piloting with tissue engineering companies for organ regeneration and wound healing with stem cell banks and clean meat companies. We're working on INDs and studies in neuroprotection and nerve damage, osteoarthritis, pulmonary fibrosis, long-term COVID damage, and also in some mechanisms of stroke and heart attack damage. We've done over 60 procedures and have over 4 million in contracts and LOIs. And we anticipate our 510K clearance coming by early Q1 of 2022. And then it's productions, distribution, and scaling. We have major distribution deals lined up and expect to hit 300K MRR shortly after we get our 510K. I did my undergrad in electrical engineering at Stanford and a master's in biotech from Cal State. And our COO, Rob Kent, has brought 10 Class 2 medical devices to market. And our CMO, Michel Haddad, he grew up in the medical industry and he knows how to close doctors. We're Y Combinator backed and Stanford StartX alums, Brian, and just closed 2.1 million in oversubscribed funding and planning for our Series A in 10 to 12 months. So I welcome follow-up conversations, and I really hope to see you again in 1,000 years and maybe on a basketball court if they still play it. Thank you.
Great. Thanks, John. Any comments or questions from the judges? As everybody comes back in.
I would just say thanks for the shout out, John. As you, I'll probably stay quiet mostly, but as other judges get their questions in. But as you know, I mean, we've taught. I used to run StartX Med's program for a little bit last year, so I'm more intimately familiar. So I'll probably stay quiet for a little bit of this while other people speak.
Sounds good.
So, John, have you done any studies on the viability of the cells after they're isolated? How do you know that those cells are viable? And then what are the different populations of cells that you're isolating? Are you keeping them all together after that? Are they being fractioned after the isolation? What's the protocol?
Okay, so we've done hundreds of isolations in the lab looking at viability. Typically, we do it head-to-head against a collagenase digestion. And what we found are usually viabilities in the 90% range, very high. We compare that to collagenase, the digestions that we do using 0.1% collagenase type 1, and those are usually in the high 80s and 90s. So our viabilities are similar. What we get is a stromal vascular fraction. So there's a number of different types of cells. There's parasites, a lot of different kinds of regenerative cells in the mix. Mainly, it's the debris and the adipocytes that are taken out. We've plated these cells. We've done extensive flow cytometry analysis. We've done hundreds of plates of cells. So we know they're viable. We know they plate. And we know from the cell markers that we look at that we're getting a solid population of cells. And when we do side-by-side comparisons, when we see that 1.5 to 2.5x in the cell yields, we also see that on the plate. Our cells are proliferating and the plates reaching confluence much more quickly than cell plated with collagenase or collagenase treated cells. We have some studies coming up to look at the secretome and the transcriptome, so then we can see exactly what's coming out of these cells and do some really in-depth comparisons.
Got it. So is it fair to assume that you're looking at markers to actually ensure pluripotency, the way that you are looking at the cells to actually make sure that they are true pluripotent stem cells?
That is accurate, yeah. So the stem cell markers that are known or typically used for cultured cells are the CD73, 90, and 105, and we verified the existence of those on the plates. 97% purity when we culture the cells out for the plastic adherent cells.
Thank you.
You're welcome.
Yeah, I was hoping Ryan was going to ask the question about the comparison with Sayenza, so I guess I'll do that. Yeah.
All right.
Yeah, seems like a simple application with them.
Seems like, say again?
No, I'll just let you go.
Oh, yeah. So we also have the ability to do fat grafting. And so that's a 510K path that you see is coming in either end of this year or early next year, and then we're off to market. As far as like our speeds, I know Derek mentioned that they take about 15 minutes. Our processing is 3 minutes. There's no one that even comes close. Our yields are 1.5 to 2.5 times higher. There's no one that comes close. When we compare it to any other mechanical methods out there, they are down. Okay, so we're 1.5 to 2.5 times higher than collagenase. We look at mechanical methods that are out there, they are usually around 30% or less compared to collagenase. So that's how much better we are than other mechanical methods. And we're also that much better than collagenase, which in the clinic, the physician usually takes up to 3.5 hours to do a single patient. So when we walk in and we say, hey, look at what we got, they're like, boom. If they're already using adipose stem cells, it's fast. There's additional regulatory stuff coming down the pipe. And so that's why we've got all these INDs lined up.
I guess since it's a little quiet out there, what just to perk us up a little bit, where do you see the market here? Do you see, what specialty types, and do you see companies, the companies in the stem cell space buying this, or do you see the phase 1, phase 2, phase 3 sites buying this, or who do you envision all the, what's the future of this space 10 years from now?
Oh, 10 years from now, it's going to be awesome. So those spaces that we talked about in the clean meats, in the tissue engineering, in the clinic, point of care, it's going to be widespread. It'll become standard of care. There's going to be a lot of stem cell banking. We've got stem cell banks that are clients that we're piloting with. Tissue engineering companies are huge. We love that space. That we're working with some companies that are doing wound healing, also liver regeneration. So basically, what I see, and that's what excites me so much, is like, hey, I am going to be able to be 1,000 years old because I'm going to be able to get my own cells and then replace organs, don't have to worry about rejection. And this space is going to blow up. There's another application that mesenchymal stem cells are also highly more efficient in reprogramming into induced pluripotent stem cells. So that's another channel that we're also exploring, 25 times higher programming efficiency.
I could throw a softball question out there, which might benefit the audience. You went through Y Combinator. A lot of the founders aspire to going through that. That was, I believe, spring 2019 batch.
Winter 19, yeah.
Yeah, and so we'd love to hear about your experience in that program.
Oh, I'm going to have to throw in StartX as well because Ryan's here. So both of those accelerators are amazing. I'll tell you, one of the huge things about it is the community and the support from the other founders, and also Y Combinator, specifically since you asked, has a lot of mentors, and we're able to continue to connect to them. It's super intense. It's a really great way to connect. I mean, I'm part of the COVID-19, we're part of a COVID-19 task force up at Stanford StartX, and then we're also heavily involved with the Y Combinator community. And so these founders are amazing. They offer just such awesome support throughout the entire process. You can ask any question you want, that somebody has been through it, and somebody can tell you how to handle just basically any situation that comes up. Yes.
So
Right.
real quick question, your 510K covers all the different use cases we talked about, from fat to the cellular application that you talked about. Like will it be a tool to be able to use in all these different use cases we've been discussing?
Not with the 510K. The 510K only covers lipoplasty and fat transfers. So that's what they have predicate devices for. Stem cells have been a thorn in the side of the FDA to figure out how to regulate, and I think they're finally figuring it out, which is great. We've got some really amazing streamlined designations that we'll be able to go through with FDA to accelerate approvals for therapeutic applications. However, those are not part of the indications of the 510K. 510K gets us out to market. Yeah. Are you muted?
You're on mute.
Kwame.
You're mute.
That $2 million that you have booked or in the funnel, are they saying as soon as you get the 510K, we'll cut the PO and let's roll? Is that kind of the scenario?
Yes, we actually have some larger contracts with some really huge distributors that want to replace somebody they're carrying now, where they're moving hundreds of thousands of units annually. They want to replace them with us, and that's pending our 510K, and that's a much larger deal than the 2 million.
You raising right now?
We just closed 2.1. There are people that are still wanting to give us money, so depending on who it is, we'll I mean, who says no to money?
Depends what it's attached to, I guess, right?
Yeah, that's true. Yeah.
I love it.
Yeah.
Well, great. Great job, John. Any final comments or suggestions or questions for John?
I just had a question to go the opposite direction from diagnostics. Do you envision going RUO, manufacturing RUO product as well? I mean, I can see the market in research use only space, like huge academic purchasers, because I know I come from manufacturing where you had primarily RUO that I was trying to take IVD. You also can expand your market going RUO.
Yeah, we've got a lot of university collaborations as well, and so that's definitely a market that is open to us that we're active in. It's not our target. I mean, it's not where we have our driving focus. However, it's and we did have some inquiries from some of the big instrument manufacturers that are in laboratories research.
John, what do you anticipate the cost of the device to
Great
be?
job, though. Thanks.
Yeah, you're welcome. I'm sorry?
What do you anticipate the cost of the device to be?
Oh, well, we know it's kind of confidential. However, we can tell you that our margins are in the 90% range.
Great.
Thank you.
Great. And we are up on time. So judges, thank you so much for the great questions. John, great job. Congratulations on all the success and best of luck.
Thank you, guys.
Thank you. Great. Okay, moving right along to our last presenter for today, Veriskin and Marianas. You with us?
Yep,
Veriskin?
I am here.
Great. Feel free to share your screen and start when you're ready.
Excellent. Well, hello everyone. My name is Marianas Kakisfilis. I'm a CEO of Veriskin, a medical device company dedicated to facilitating and improving the accuracy of skin cancer diagnostics. Skin cancer is the most common cancer in the US with over 5 million cases annually. Unfortunately, skin cancers are very difficult to distinguish from benign lesions, which leads to two problems: thousands of lives lost and millions of unnecessary referrals and biopsies, resulting in $3.2 billion of avoidable cost. It has been established that early skin cancer detection can save lives and reduce costs. Our solution is TruScore, a non-invasive, handheld, low-cost, patented device for non-expert clinicians that rapidly determines if a suspect lesion is malignant. Clinical adoption of the TruScore device would benefit all stakeholders, patients, providers, and payers through improved patient outcomes and reduced cost. Veriskin's non-imaging-based technology and AI algorithm works by detecting structural and functional vascular abnormalities associated with cancer-induced pathological angiogenesis, which is a well-established early hallmark of cancer. Our pilot clinical studies on 125 biopsy-verified lesions have demonstrated 100% sensitivity and 94% specificity. Clinical adoption of TruScore would greatly improve diagnostic performance of non-expert clinicians. Inherently higher diagnostic information content of the TruScore's hemodynamic test as compared to competitors' imaging-based technologies offers superior accuracy at lower cost and with no skilled user requirement. Veriskin's business strategy is based on a virtual razor blade model. We will focus on distribution to primary care providers and dermatologists through a sale of a device and a software enforced per-use fee. The total addressable clinical markets in the US and EU are estimated at $2.4 and $5.3 billion. FDA has granted TruScore the breakthrough device designation status, which entitles Veriskin to automatic Medicare reimbursement upon regulatory clearance expected in 2023. Veriskin intends to pursue FDA pre-market approval. The company plans to exit via pre-market sale to a larger strategic player also in 2023. Veriskin has finished technical development of the production device and set up a certified manufacturing line. Veriskin's IP portfolio includes issued patents and pending applications. The company has been supported by 2 grant awards from the NIH, totaling at $3.9 million. Next steps in the commercialization efforts include a final pre-pivotal algorithm training study this year, followed by the FDA's pivotal trial in 2022. The company is seeking to fill out the first tranche of the $2.5 million seed round to support FDA CE regulatory clearance. We have raised just over $1 million up to date. Company is looking for additional co-investors into this round. Veriskin has been founded by 3 experienced scientists and serial entrepreneurs with successful track records on multiple exits. To conclude, the adoption of TrueScope by primary providers and dermatologists will improve patient outcomes and reduce costs. Thank you, and I'm open for questions.
Great, thank you, Marianas.
Mhm.
As our judges come back on, would anybody like to kick it off with the first question or comment?
Marianas, when you were looking at the sensitivity and specificity, what type of lesions were you looking at in terms of, obviously we can say a melanoma is quite easier to diagnose compared to other types of skin cancer. So how was that done? What lesions did you look at and what were the numbers?
Okay, so we had a mix of lesions, including basal cell carcinoma, squamous cell carcinoma, and melanoma, and variety of benign lesions like actinic keratosis, seborrheic keratosis. You take it. Whatever was misdiagnosed by the dermatologist, and we got it as a false positive. So, but regarding your comment about melanomas are the ones that are most difficult to diagnose. It typically nowadays takes 15 biopsies just to catch one melanoma, because skin cancer, especially a melanoma, is like it's like an iceberg. What you see on the top visually, it's just not enough information to diagnose accurately. Therefore, physicians tend to err on the side of cautions and just cut it out. That leads to enormous costs to the healthcare, and of course, it's no benefit to patient who had a benign lesion.
Thanks.
What study did the FDA say you need to do for your breakthrough designation? And can you say a little bit about how many sites you're going to do it at, et cetera?
Yeah, so the breakthrough device designation was awarded last year, and it doesn't present any requirements to us on the studies. It's just indication of a novelty of the approach and the need and the market need. All right, so that's the criteria why it was awarded to us. Now, as far as a clinical trial, we'll need to do trial on around 2,000 patients, and so around 20 sites per FDA guidance. Probably half of them in the US and the rest in Australia, where the incidence of melanomas are higher. And the cost is projected somewhere between $2.5 and $4 million, of which most likely half of which will be paid by the NIH from additional grants that we are currently seeking and the existing grants at the moment. And it will going to take, right now, based on the full quotes from a CROs that we contacted, and we talked to 4, so from 9 to 12 months, because this is such a common cancer, right? So the patient recruitment rates are really high, and it's non-invasive trial. Therefore, you don't have to do patient follow-ups, because you just got to get a biopsy results to determine diagnostic accuracy.
And you may have said this. Did the FDA indicate whether this will be a PMA or a de novo?
No, it's a class 3 PMA.
Okay, thank you.
Yep.
Can you talk more about exits? And first off, thank you for the presentation, high unmet need. And I thought your presentation was phenomenal. I read it in the prep. One area that I want to dig in on a little bit was maybe the comps in this space, particularly in melanoma diagnostics. Has anyone really done it successfully and exited successfully yet, specifically in this need area? Or is it still such a high challenge that nobody's really had success? I saw on your slide 10, the sensitivity specificity is kind of ranged.
That's right. Yeah. So there's been a number of failures in this space, but there are some current successes. For example, San Diego-based Dermtech, which specializes. It's not technically it's not a device. It's a non-invasive patch for melanoma diagnostics, and it's clear lab. Essentially, you peel some cells from the lesion and you send them for the genomic analysis, and this way they're able to diagnose melanoma or non-melanoma. So they did a reverse IPO. I think they had $60 million, raised $60 million in that, and so they raised also significant funds from the private funds. And so they're doing well right now. They're commercializing it. And the most important thing that happened, I believe last year, the Medicare granted them a $750 reimbursement per one lesion. So, which bodes really well for this sector and for everybody who is trying to do this. There's no reason why for example, Veriskin's test wouldn't get a similar reimbursement, because we essentially do the same thing.
Thank you.
Mhm.
Any other follow-up questions or comments? Marianas? Just checking the chat box. Okay, well, great presentation, Marianas, and congratulations and best of luck on all the opportunities that lie ahead.
Thank you very much. Thank you.
Thank you.